All right. Good morning, everybody. Lots of people in the crowd today. I love it. We got lots of people online. Let me dismiss this. Thank you all for attending today. We've got a really packed agenda, but an exciting topic for us to discuss, which is the coordination and support for the response to and recovery from a biological incident that could occur in the Commonwealth. So thanks again, everybody, for joining. The training team is going to be putting the attendance record in the chat, so please fill that out as you go through the training today so we can really capture the audience for both the reporting for the Virginia Department of Health, but also for VEST training records. So with that, I'm going to turn over to Bob Moskov, our emergency preparedness lead here in the VEST, a valued member and partner of the VEST, to introduce his team.
Good morning. A little historical or hysterical perspective, depending on your point of view. The VDH established its emergency preparedness and response program in 2002 as a result of 9/11 and the anthrax attacks that happened almost simultaneously. VDH receives two significant grants from CDC and HHS. One is the Public Health Emergency Preparedness Grant, and the other is the Hospital Preparedness Grant. And the Office of Emergency Preparedness within VDH, which I run, manages those two grants. So our Office of Emergency Preparedness has been around since October of 2022. My partner in crime, Susie Silverstein, who most of you know, and I have been co-leads essentially in that since the very beginning. We were originally founded as a bioterrorism response entity, and my original title, and I use this a lot, but my original title was the State Bioterrorism Coordinator. And so when I came into the job and I was being interviewed, I asked, was I going to actually coordinate bioterrorism for the Commonwealth of Virginia? They all agreed that that was a misnomer, so we quickly changed it to an all-hazard ESF. About the same time NIMS came in, we all adopted ICS, and we started churning and burning as an all-hazardous response entity. I was joking with Michelle that most of the people in VDH understood things like diseases and environmental threats and drinking water threats, and we knew that there would be responses on the health side to a hurricane. But we hadn't drilled or anything for what is our role during a hurricane. So we did a very robust exercise on hurricane response. As a result, got Hurricane Isabel within a month. So fast forward, in 2019, we said, you know, we've had a lot of threats about pandemic influenza. We've had bird flu, we've had swine flu, we've had the avian influenza started way back. In fact, what many of you may not know is when the threat of avian influenza started pre-2010, we started looking at two separate parallel courses, one for infrastructure, which was being led in the state by VDEM, and one for health, which was being led by VDH. So, because if we had a pandemic, the infrastructure, as we now know, would be clobbered, and it was during COVID. I say COVID, and so we had those two. Since then, we've become enlightened, and we know that there really is a reason that we have a VEST, and there really is a reason that we do consolidated and coordinated initiatives. So we did an exercise in 2019, and it was a pandemic of influenza that started in Wuhan, China, go figure. And we came up with this scenario. Jonathan Falk and the EPI team put together about a 6-month lead up, an EPI surveillance and investigation that led up, and we went around the state and started looking at how would something like this beginning to transpire. And it culminated in a big exercise in 2019, October. And as you know, in February of 2020, we got COVID. So I don't know if we're going to, I don't know if this portends that we're causing the next great pandemic. VDEM and VDH have been partners, and I'll add VHHA, the Virginia Hospital Healthcare Association, actually preceding 9/11, but really after 9/11, it solidified. If any of you worked at other states, this relationship does not exist in other states. This partnership does not exist in other states. Things like a vest, where we all come together to do Friday schoolhouse or exercises or the meetings that we do judiciously or the Thursday get-togethers. This doesn't exist in other states. Other states have very formulaic stovepipes, and sometimes they attempt to bridge those. We don't have that situation in the Commonwealth. And one of my roles is as the best liaison officer for the Virginia Department of Health, and we run ESF 3 and 8. 3 is public works. Why do we do public works? Because 2 reasons. The state coordinator said, "Would you do public works?" And the commissioner said yes, and said, "We're going to do we're going to do ESF 3." And now Jessica eats, lives, drinks ESF 3 all the time, but she remains within the rubric, the house of the Virginia Department of Health. We've had, as I said, these different influenzas. H9N1, which was swine flu, was a big one about 2010. And it became sort of a forecast of what might happen, but nothing like the COVID. We've done Zika, we've done Ebola, we've done Mpox, and so many others. And we look at annual influenza every year. We deal with overdoses. Oh, by the way, we run an incident management team in overdoses, and we saw a 34% decline in overdose deaths within the last year because of the efforts of our IMT and works of the Secretary of Public Safety and Homeland Security and the efforts to thwart fentanyl. That's a big deal. So it's, we see this as a public health threat as well. Part of our requirement for getting the money to do these 2 grants that I mentioned is that we do exercises. And so even though the threat of causing another pandemic by having this exercise is there, we have certain requirements that we have to go through. So we're starting at the baseline with what we're doing here. And it will culminate in something down the road. For those of you who weren't here in COVID, we deal with hurricanes and we deal with earthquakes and we deal with floods and we deal with things. Most of these events come and go within a relatively short period of time. The stuff that the VDH deals with is the gift that keeps on giving. We're still dealing with the aftermath of COVID. And we're still dealing with some of the aftermath of H9N1. And we deal every year with annual influenza, but it's the novel influenza. And I'll mention the partnership that we have with our state laboratory has been key all that time as well. So this is a partnership of partnerships. So the grant requires that we have this familiarization with our members in the other studies in the other fields of emergency preparedness and response. And in other states, when they're doing this, they're exchanging business cards perhaps for the first time when they do this. And for us, I think we know everybody in this room. We've gotten together either on the screen or in person for years. So part of the response to a large influenza and perhaps a pandemic is the epidemiological lead up, the management, and then there's this thing that health departments don't normally do, and it's the operationalization and logistics support for response to a COVID. So harken back to February and March of 2020, and somebody said, there's going to be a big need for personal protective equipment here. What kind of personal protective equipment do we have in the barn? We had a lot of leftover stuff from H9N1, from swine flu in 2010. So we went to the barn and we broke it out, and we found all the elastics in our masks were deteriorated. So we actually hired a company to re-Band-Aid, put new elastic, if you will, on the stuff we had. We went to the strategic national stockpile, which was the intent was for that would take care of all of our medical countermeasure requirements, including personal protective equipment. And they said, okay, we'll get it. And they weren't prepared for all states and territories to have this type of a situation simultaneously. And that's what we had. So the stockpile was non-existent. So I remember coming here, having a whole lot more because everybody's saying, what the hell is COVID? And this place was actually full to the gunnels. And we gave a threat brief. And then we went over to VDEM headquarters the next day to start the operationalization of the response. And the first thing that was asked was personal protective equipment. And we were on a call with CDC at the time, and they said, we can get you a couple of thousand of these and a couple of thousand of those. And they said, we got about 8 million people living in the Commonwealth. If this really is a pandemic and it grows like the exercise that we had in October said it would, we're going to need a lot more than that. And they went. So Jeff Stern was the state coordinator for the Department of Emergency Management at the time. The governor called VDEM and VDH up to his office and said, this will be an all of government response. And he wasn't kidding. He said an all of government response, quote unquote. And we are going to use incident command. We're going to use, I think it was called the VERT, the Virginia Emergency Response, rather than the Virginia Emergency Support Team at that time, and it's morphed since then. So VDEM and we started the search for personal protective equipment, and they primarily VDEM went out and started looking and securing stuff from all over the world. And it was flown into Chicago and trucked from Chicago to here. And then we did a distribution matrix and a prioritization plan on how it would be distributed. The hospital said we want it all. Nice. But the jurisdiction said we needed to maintain continuity of government, continuity of operations. So so we actually met here almost daily with our masks on, right? Every day, whiteboard, and said, okay, and we came up with a matrix and a prioritization. And we made it work. And eventually we became, I won't use the word flush, but we came to have as much as we needed by the end of the pandemic. We continue to maintain a very robust stockpile in the VDEM. What's the term? We don't call it a warehouse. We call it the Logistics Service Support Center, I believe. But we have like 1 million antigen tests over there, and we have a lot of PPE. The stuff with PPE that we learned before was it deteriorates, and everything that's a medical countermeasure has a shelf life. And even if it's not useless, according to the food and the FDA, they say you can't use it unless there's some kind of declaration that extends the shelf life, that sort of thing. So this is the ground truth that we're beginning to look at. We're going to talk about the epidemiological buildup and surveillance and investigation. But eventually we'll morph to the operationalization and the logistics support for something like this. Now, in my mind, if somebody isn't dead, injured, or sick, it's not an emergency. That's the way my mind works, but I'm an health. You know, there's a whole lot of other things that happen in emergencies from the perspective of emergency management, like property and infrastructure and everything like that. But from health's perspective, if somebody's not dead, sick, or injured, it's not an emergency. So that's the way we look at it. But we recognize the partnership and all the other things that go into this. Why else would we have 17, I think, or 18 emergency support functions? So thanks to VDEM for having this type of a training, because of everything else we've done as a VEST, nothing else but COVID, a disease, lasted for 3+ years and absorbed and kept us as a VEST, an active VEST, for 3+ years. And no hurricane, no flood, no earthquake, no nothing kept us. So don't dismiss this when we look at the THIRA, the threat hazard identification and risk analysis. Make sure you keep infectious diseases and the threat of a pandemic or an epidemic up there in your thoughts. With that, I'll turn it over to Brandy to start us off, and thank you very much.
Good morning, everyone. For those of you who don't know me, my name is Dr. Brandy Darby. I work within the Office of Epidemiology at the Department of Health, and my role is as the director of our Division of Surveillance and Investigation. Within that division, we have both our respiratory disease epidemiology team and our zoonotic disease epidemiology team, both of whom are here today. And are obviously very involved when we have these novel influenza patients come up and we're trying to do rule outs to see what's happening. We do get those from time to time. I really appreciate everyone who's come together, particularly those who've put together presentations and content to provide this foundation educational background about flu that will help inform some of the future exercises that we go through in future grant years. What I hope you'll see today is that, one, flu is ever changing just by the nature of what it is. It's an RNA virus, and those viruses are notorious for evolving and changing, sometimes suddenly and in unprecedented ways. So this is something that repeatedly keeps us on our toes. And because of that, we have robust surveillance to continuously monitor these viruses that are in circulation. And the other thing that I hope that you see is really the multidisciplinary One Health approach that it takes to do flu surveillance properly. So you'll see not only our epidemiology teams, our laboratory teams and testing abilities here in the Commonwealth. We have our animal health partners who are with us, emergency health partners, healthcare coordination. So a number of people. We can't do this work independently. It takes a lot of different backgrounds and perspectives coming together to make the magic happen. I'm not going to tell you anything that you probably don't already know. If you're tuned into the news, you've seen that flu changed a lot in the last year. We've known avian influenza viruses for a very long time. That's the natural reservoir for a lot of our influenza A viruses. Starting in January 2021 or 2022, we started to see this new H5N1 virus in birds in North America. That was new for us. We've had these flu introductions. The viruses normally circulate within wild birds, and then we'll get spillover to domestic birds from time to time. But this persistent spillover that's been happening for 2 or 3 years now at this point is unprecedented and is the largest animal health emergency that we've seen in the U.S. Every time that this virus replicates, it has the potential to change. Last year, we saw spillover from birds into cattle, so that was again new. It's infected a number of different mammalian species, so we've seen it. You'll hear about different species. We've seen this in Virginia from our animal health partners a little later today. So just a lot of flu in the environment. We do, and I'll talk a little bit about some of the things that we've done in the last year as far as preparedness for any sort of potential novel flu event. And when we say novel flu, we're meaning something that's not sort of the typical seasonal influenza viruses that normally circulate in human populations. When we had the original spillover of flu into cattle, these farmers, we didn't know about PPE preparedness, and so we partnered with the Office of Emergency Preparedness to help push out supplies of PPE to dairy producers, poultry producers, and slaughterhouse workers across Virginia. Our cross-cutting epidemiology team and zoonotic disease team had a really strong role in making that distribution happen. We've also sent out a number of communication messages to healthcare providers in Virginia, encouraging them to have a very low threshold. So think about flu, test for flu, especially in people who've had some of these animal exposures, even if the symptoms are very mild. And I want to say thank you to our lab. They've tested a large number of flu samples for us so that we can quickly, rule in or out if some of these H5 or novel flus might be playing a role in human illness. The goal there being to very quickly identify any human cases if present. We've also sent out communications to providers to try and increase our flu A subtyping, which gives us a better idea of the viruses that are circulating in communities. The lab was quickly flooded when we sent out this request, and so we were definitely successful in that effort. And, the other thing about flu, I think we sometimes just not we like public health, but general public can sometimes get a little bit complacent because we think, oh, it's just the flu, it's not that bad, and people get over it. We do see severe outcomes with flu, and this particular flu season was particularly severe, both with the number of people, but also with the severity of illness. We had a number of children, I think 5 or 6 throughout the Commonwealth, who died of flu this year. We also saw some severe complications, including flu-associated encephalopathies and acute necrotizing encephalopathies. So just really severe outcomes. So definitely not a disease to be complacent about. We're excited to help kick off this series of training events, and I hope you enjoyed it. Okay, thank y'all.
Thank you, Bob and Dr. Darby. For those of you that don't know me, my name is Aaron Kiesacker. I'm the exercise officer for the VDH Office of Emergency Preparedness. So I am the one that is tasked with leading this endeavor from start to finish. So thank you, everyone that has attended or that is attended. Bob's already covered this very well, so I'm going to save most of my comments for the end. And I will skip right to the beginning of our content. I'll pass it off to Lisa. You coming up here? All right. Sure.
Awesome. Thank you. All right. Hi, everybody. My name is Lisa Salad. I'm the Respiratory Disease Program Coordinator for the Office of Epidemiology. And I'm going to go into a little bit about the background of influenza, as well as some virology. So first of all, let's just take it back to what is an influenza virus. So influenza viruses cause contagious respiratory disease, and they're part of the family Orthomyxoviridae. They can be categorized into 4 different types. That's influenza A, B, C, and D. You're probably most familiar with A and B because they cause seasonal epidemics of disease in people. That's known as the flu season, which is typically in the winter in the United States. Influenza C viruses generally cause a more mild illness, and we don't really have reported epidemics for that, which is why you don't hear much about it. And then influenza D viruses mainly infect cattle and are not known to infect humans. So for the purposes of this discussion, we're going to talk mainly about influenza A viruses, which are responsible for avian influenza. So influenza viruses have a segmented negative sense single-stranded viral RNA genome contained within the nucleocapsid. Influenza A and B viruses have 8 genomic segments, and these genomic segments code for both structural and non-structural proteins needed for viral replication. The outer surface of the virus that you can see here, the viral envelope, contains 2 important transmembrane glycoproteins, hemagglutinin and neuraminidase. Hemagglutinins are shown in the image as the purple taller stalks, and the neuraminidase are shown as the pinkish clover-looking stalks. Influenza A viruses are subtyped according to the antigenicity of these glycoproteins. There's 18 different hemagglutinin subtypes and 11 different neuraminidase subtypes, and these make up what you're used to hearing influenza A virus is referred to as. So H3N2, for instance, means those particular hemagglutinin and neuraminidase, and H1N1, H5N1, et cetera. So influenza viruses are constantly changing, as we've mentioned, and these changes can either happen slowly or they can happen suddenly. When they happen slowly, that's called antigenic drift. So this happens every flu season from October to May in the United States. Different strains of seasonal A and B influenza are passed from person to person, which causes illness. The drift that happens there happens between small changes or mutations in the genes of the influenza viruses in the people and then passing it back and forth. These slow and constant changes to the flu virus are the reason that people need to get a flu vaccine every year. However, sudden changes to the flu virus are called antigenic shift. Shift is an abrupt major change in an influenza A virus, resulting in a new or novel flu virus that infects humans. This can occur when the genetic material from two viruses combine to form a virus with a new subtype or a mix of genes, including some from animals. Most people do not have immunity or protection from novel flu viruses because they're different than seasonal flu and our bodies haven't seen them before. An example of this occurred in the spring of 2009 when a new H1N1 virus with a different combination of genes from pigs, birds, and humans emerged in people and quickly spread. In summary, flu viruses undergo antigenic drift all of the time, while antigenic shift happens more infrequently. And then the hallmark of influenza viruses that provides the ability for these shifts is the segmented RNA genome, as Dr. Darby was referring to. Because of those 8 genomic segments present in influenza A viruses, if an individual is co-infected with two influenza viruses at the same time, there are theoretically 256 possible genetic combinations that could arise. This process of antigenic shift or reassortment is important to driving influenza evolution and species spillover events. Subtypes of influenza A viruses that routinely circulate in people include AH1N1 and AH3N2. Influenza B viruses that circulate in people are broken down by lineage. So B Victoria is the only influenza B virus currently circulating in people. You may have previously heard of B Yamagata, but that's interestingly not been seen since 2020 or 2020. Influenza A virus subtypes and influenza B viral lineages can be further broken down into clades and subclades. For influenza A viruses, these clades and subclades are based on the similarity of their hemagglutinin gene sequence. You may have heard that the H5N1 virus in dairy cows has had 2 clades detected, B3.13 and D1.1. Both of these are H5N1 influenza A viruses and are antigenically similar but genetically different. This means that a host's immune response, or the antibodies inside the host, elicited by infection or vaccination with one of the viruses, will recognize and neutralize the other virus, thereby protecting against the other virus. All right, so to bring it more to avian influenza now, so of course we're going to be talking about A viruses. Avian influenza refers to disease in birds caused by infection with avian or bird influenza. These viruses occur naturally among wild aquatic birds worldwide and can infect domestic poultry and other bird and animal species. Wild aquatic birds include water birds like waterfowl such as ducks, geese, swans, gulls, terns, and shorebirds such as storks, plovers, and sandpipers. Wild aquatic birds, especially dabbling ducks, are considered reservoirs or hosts for avian influenza viruses. Wild aquatic birds can be infected with influenza A viruses in their intestines and respiratory tracts, but some species, such as ducks, may not actually get sick. As the natural reservoir for influenza A virus diversity, all but 2 known subtypes of influenza A viruses have been detected in wild aquatic birds. Subtypes H17 and N10, and H18 and 11 have only been found in bats. When we look at this image here, we can see how complicated viral ecology and how influenza is significantly relevant to public health, wildlife health, as well as the agricultural industry, as infections not only impact many species, but cause infrequent interspecies transmission. It's generally uncommon for people to get influenza viral infections directly from animals. However, sporadic human infections and outbreaks caused by certain avian and swine variant influenza A viruses have been reported. And of course, given that this graphic here is from a 2016 publication, we know that we're currently experiencing H5N1 linked to dairy cattle, which would be a new addition to this viral ecology picture here. All right, and again, occasionally influenza viruses circulating in wild aquatic birds can cause human infection, often through intermediate hosts such as swine or poultry, or more recently, dairy cattle. Usually infections are isolated to direct contact with the infected animal and do not sustain human-to-human transmission. However, upon viral mutation or reassortment, the zoonotic virus can adapt to human hosts, which caused major epidemic and pandemic events in the last century. Epidemics are large outbreaks of disease, and pandemics are large outbreaks that spread rapidly at a global scale. With each of these major disease events, it is thought that there was an initial spillover event from animal populations in close contact with people, which is usually avian, but as we saw in 2009 with swine. Spillover events, they happen occasionally from animals to people, but don't often lead to widespread disease in people. When the influenza A virus adapts in such a way to be able to transmit efficiently from person to person after the initial spillover event, that's when we start to see these larger outbreaks that can pose a more serious threat to human health. So you're going to hear a little bit more about the flyways later. I'll leave that to the experts, but I just want to show you this for an idea of how migrating birds can spread and share virus at staging, stopover, and wintering sites. Influenza viruses or avian influenza viruses are thought to be shared where migratory flyways overlap. And you can see here there's very significant overlap between those flyways. And that leads to co-infection, reassortment, and it can lead to the emergence of new viral subtypes. Most times, wild aquatic birds infected with influenza A viruses don't actually get sick, as we've mentioned. However, influenza A viruses are still very contagious among birds, and some of these viruses can sicken and even kill certain domestic bird species, including chickens, ducks, and turkeys. Influenza A viruses are classified into the following two categories: low pathogenicity influenza, or LPAI viruses, and highly pathogenic avian influenza, or HPAI A viruses. The categories refer to molecular characteristics of the virus and the virus's ability to cause disease and mortality in chickens specifically. A low pathogenic avian influenza causes either no signs or disease or mild disease in chickens and poultry, such as perhaps some ruffled feathers or a drop in egg production, while most avian influenza A viruses are low pathogenic and cause few signs of disease. In poultry, some low pathogenic viruses can mutate into highly pathogenic avian influenza viruses. These highly pathogenic avian influenza viruses cause more severe disease and high mortality among the infected poultry. Oops, sorry. Moved my slide. Only some avian influenza viruses are classified as HPAI viruses, like H5 and H7. While most H5 and H7 viruses circulating among birds are LPAI viruses. All right, so clinical signs in a poultry flock can appear between 2 and 21 days after their initial exposure. There's no significant treatment available for influenza A infected birds. HPAI can rapidly cause up to 100% mortality in affected birds. There's also a high risk of transmission of HPAI to neighboring flocks. Therefore, poultry flocks with a positive PCR diagnosis of H5 or H7 with clinical signs of HPAI and a mortality of 30% or greater require immediate depopulation within 24 hours and on-site disposal of the affected flock. USDA's Defend the Flock education program encourages biosecurity practices to keep poultry healthy and reduce the risk of avian influenza and other infectious diseases. Examples of biosecurity practices include wearing personal protective equipment or clothing and shoes that are only used when caring for poultry, removing litter, manure, and other debris, washing hands thoroughly with soap and water, cleaning equipment and vehicles before and after they come in contact with your flock, and knowing signs of flock illness and who to report it to. All right, and to dig a little more into novel A viruses. Novel A virus infections include all human infections with influenza A viruses that are different from currently circulating human seasonal strains. So those would be the H1s and H3s. Novel viruses include those that are subtyped as non-human in origin and those that are not able to be subtyped using standard laboratory methods and reagents, which you'll hear a little bit more about later. In 2007, human infection with a novel influenza A virus became a nationally notifiable condition in the United States. Rapid detection and reporting of human infections with novel A viruses against which there is little to no pre-existing immunity is important to facilitate prompt awareness and characterization of influenza A viruses with pandemic potential and accelerate the implementation of effective public health responses. Again, H1 and H3 subtypes originating from a non-human species or from genetic reassortment between animal and human viruses are considered novel viruses. For example, human infections with an influenza virus that normally circulates in swine and not people are called variant viruses. They can be denoted by adding the letter V to the end of the virus subtype designation. Human infections with A H1N1V, H1N2V, and AH3N2V viruses have been detected in the United States. You'll see here confirmed human novel influenza A virus infections over the past 15 flu seasons. H3N2 variant viruses, denoted here in yellow, were identified in a multi-state outbreak, first identified in 2011 and associated with swine contact at agricultural fairs. More recently, H5N1 viruses, here in red, have caused human infections associated with exposures at infected dairy cattle and poultry. When novel flu is detected in Virginia, VDH supports response efforts with a focus on protecting human health and preventing additional human illness. The steps of a public health investigation for novel flu typically include working with the state and federal animal health partners, agricultural industry partners, and other affected groups to identify potentially exposed people to birds, animals, and their byproducts. Interviewing these people to understand their exposure risk and to help them monitor their health for any signs of illness, as well as provide education. Quickly coordinate any testing for people who develop flu-like illness or eye redness and irritation or conjunctivitis or other relevant symptoms. And then if an exposed person does develop symptoms, VDH recommends rapid medical evaluation and flu antiviral drugs to treat a suspected infection. Flu antiviral drugs may be offered to people who've had exposure and did not wear the recommended personal protective equipment or who had a breach in their PPE. I know there's going to be a little bit more into this later when Dawn really digs into the field response, but to give a brief overview before some of the other discussion, one of the first steps of an epidemiologic investigation is typically always going to be getting more information. VDH is typically notified in one of two ways about novel flu events that require public health follow-up, one of which would be routine reports from CDC over a secure platform called EpiX with names and contact information for federal responders who may have responded to an event from USDA APHIS that supported response efforts out of state, but are Virginia residents and returning in state. And then the other way is we also expect to receive reports from VDAX or DWR in the case of animal health events in Virginia when H5 or novel flu virus is detected. Local health departments also receive reports from clinicians or patients concerned about potential illness or exposure to novel flu. Again, the first steps usually involve additional information gathering. This usually includes gathering more information about the extent of the potentially exposed people. Is it employees? Is it on-site personnel? Is it visitors? Could it be household members? And then gathering information about their potentially infected animals. So how many animals are there? What species are there? When did they start feeling sick? And what did their illness look like? Are there other animals on the property? And all of their health information, like their signs and symptoms, and to gather information like whether or not they might need medical evaluation, follow-up, or testing. Next, the investigator would use epidemiologic criteria to determine who would be considered exposed. The novel influenza A virus case classification offers a definition for epidemiologic linkage, and guidance for H5N1 specifically offers more detailed examples of activities that qualify as an exposure. Generally, exposure is defined as close contact with a confirmed human case of novel influenza A virus infection, or shared common exposure, such as an agricultural fair or live animal market with a confirmed novel influenza A case, or direct or indirect contact, such as touching an animal, their environment, or their raw or unprocessed animal product with animals with confirmed influenza A, or lastly, inadequate use or breach of PPE and exposure to novel influenza A virus in a laboratory. Signs and symptoms of illness are consistent with infection of novel influenza A virus infections in humans. And they've ranged from no symptoms or mild illness, such as eye redness, conjunctivitis, or mild flu-like upper respiratory symptoms, to severe illness, such as pneumonia, even requiring hospitalization. Usually includes fever as well, so that's a temperature of 100 degrees Fahrenheit or 37.8 Celsius or greater, or feeling feverish. Cough, sore throat, runny or stuffy nose, muscle or body aches, headaches, fatigue, shortness of breath, or difficulty breathing. Less common signs include diarrhea, nausea, vomiting, and seizures, and can be more common in children. Note that these signs and symptoms are non-specific and overlap with those caused by other respiratory viruses, including seasonal influenza A and B viruses. Further evaluation of symptomatic people should be performed by state or local public health agencies to assess whether testing, isolation, and/or treatment is warranted. As Dr. Darby was referring to, we really push the importance of testing, not just to differentiate the specific influenza A virus, but also to tell whether it is indeed influenza or could be another co-circulating respiratory virus. VDH provides approval for novel testing prior to submission to DCLS. Our wonderful partners there are quick in having us send specimens over, so we try to be quick in letting them know as well. For a symptomatic person, testing for novel flu would be for anyone who meets epidemiologic and clinical criteria. Whereas for an asymptomatic person, testing for novel flu would be offered when feasible following a high-risk exposure while not wearing full recommended PPE, or if they've experienced a known breach in their PPE. So something like having raw milk splashed on their face or into the eyes, or if they have close contact with a confirmed human case, such as a household member. All right, and then antiviral treatment for influenza, also known as, oseltamivir, also known as Tamiflu, is recommended for treatment or post-exposure prophylaxis of novel influenza A viruses. It's recommended that symptomatic patients and people with high-risk exposures begin oseltamivir as soon as possible. All right, and I'm going to wrap it up with a little bit more information about the surveillance activities performed at VDH for novel influenza A viruses. One of the ways that we do this is with case reporting. So as I mentioned before, novel influenza A virus infection is rapidly reportable by clinicians in Virginia. Rapidly reportable means that they need to report it by the most rapid means possible within 24 hours of knowledge. Influenza A positive results with no subtype identified or obtained, i.e. flu A unsubtypables, are rapidly reportable by all laboratories in Virginia. We also have syndromic surveillance. VDH uses syndromic surveillance to monitor data from visits to emergency departments and urgent care centers in Virginia. This is one of the fastest ways to spot changes in trends and allows us to query visit data for a wide variety of conditions, including reportable and non-reportable conditions. We've developed specific queries that look for combinations of influenza viruses or symptoms and mentions of animal exposures or certain occupations. You can also query by a set of multiple conditions and symptoms, which we do when we track trends for acute respiratory illness. This looks at diagnostic codes associated with both specific respiratory infections, e.g. influenza, respiratory syncytial virus, or coronavirus, and codes associated with general respiratory illness, such as cough or pneumonia. This can be especially useful when we're looking into something that might be happening in real time. For instance, Dr. Darby mentioned that there were more severe symptoms seen this flu season. So our syndromic surveillance platform, Essence, really helped us to really dig in further and see if we were seeing those more severe outcomes here in Virginia, and allowed us to pass that information to our local health departments so that they could follow up with providers and get a little bit more information. All right. And then next, we also perform sentinel surveillance. So that's a program we've conducted for many years aimed at better understanding the respiratory viruses circulating. We work with healthcare providers to voluntarily request that they submit respiratory specimens from patients with influenza-like illness to DCLS. DCLS tests those specimens for respiratory viruses and can further characterize any flu-positive specimens and send them to WHO reference labs for sequencing, which informs global and national vaccine composition decisions. This routine testing strategy helped identify several of the novel flu human cases in the US during 2024 with unknown exposure. Again, DCLS receives and tests all flu A unsubtypable specimens from laboratories in Virginia to confirm if they are seasonal subtypes or a novel subtype. And then lastly, VDH conducts wastewater surveillance for flu A and flu B at over 30 wastewater sampling sites in Virginia. These samples are tested and trends are tracked and compared to other indicators for any notable abnormalities. CDC has 5 sites in Virginia currently contracted to test for H5 targets specifically, and there have been no H5 detections in Virginia wastewater to date. And that is it for me. I will pass it to Ludwig. Okay.
So good morning, everyone. Just wanted to share a little bit of the activities that are performed here at the state laboratory as it reference influenza surveillance and response activities. Just a brief history for those that don't know, DCLS is a division within the Department of General Services. This laboratory was formed in 1972 with the idea of combining several different Virginia agencies under one umbrella as a more efficient and cost-effective way to perform laboratory testing. DCLS was the first consolidated public health laboratory within this country. We serve hundreds of state and federal agencies, conducting about 9 million tests per year across all of our testing programs, using about 650 different analytes. We do offer a wide variety of unique laboratory testing in support of public health, in addition to food safety, environmental protection, agriculture, and of course, emergency response. And we do have a comprehensive testing menu, some of which that are listed here, including our newborn screening, our clinical testing across immunology, microbiology, molecular biology, in doing the response of COVID. For those several years, we brought on better infrastructure for next generational sequencing. So we're able now to do WGS. We also do some environmental testing as well as commodities testing. This is just a diagram of our current organization to kind of give you a breadth of the various testing programs and subject matter areas that are consolidated under one division here at DCLS. And as you can see, we have 3 main umbrellas of testing services that are all led by our laboratory director, Dr. Denise Toney. I won't go into too much on influenza viruses because Lisa has already taken a deep dive into that, has done a great job. So I won't be repetitive for this. Again, novel influenza viruses, these are those that are causing human infection, but not considered to be any of the seasonal strains. As Lisa mentioned, they are typically classified as influenza A viruses. Of course, we understand their impact to public health because of their likelihood to cause pandemics, cause serious human illness and mortality, and of course, the ability to spread from person to person. The other thing that is important with these novel strains is that there is very little to no immunity within the human population. Human infection with a novel influenza virus is a nationally notifiable condition that does require reporting to the CDC. In the past, novel virus strains that have caused significant public health concern includes influenza AH5, which is also known as the highly pathogenic avian influenza, influenza AH7, which is considered low pathogenic, and then of course the swine flu strains H3N2 and H1N1. Won't go into this because this was already discussed through Lisa, so I'll skip past that. And then we also already talked a little bit about swine flu, its impact with public health back in like 2009, 2010, the ability for these viruses to mutate and then shift among other flu strains. To attribute a human influenza infection to novel flu virus, clinical, laboratory, and/or epidemiological criteria must be met. And some of this criteria includes, of course, symptomology. And then when it comes to laboratory testing, a positive confirmatory result using a molecular test such as real-time PCR, which is the main method that DCLS utilizes for our influenza testing. And then, of course, genetic sequence data that also indicates a novel influenza A strain. In addition, virus isolation, which is, being able to propagate a new viral influenza from a clinical specimen. Of note, from a biosafety, biosecurity standpoint, we will not perform virus isolation or any clinical specimens that are associated with either a novel influenza or avian influenza case. And then, of course, category 3 is evidence of infection, which can be denoted by increase in IgG antibodies towards a novel influenza A. So that's confirmatory. Presumptive would be a positive PCR test designed to detect a novel influenza, such as the assays that are done at DCLS, and then also test results indicating a variant influenza. And I'll talk about this a little bit more. The current assays that we run at DCLS can give us an indication of a variant, but it's not a true result that we're dealing with a variant. Surveillance initiatives to identify and monitor influenza infections, of course, require a multi-pronged approach. As Lisa mentioned, the APHI response in 2024 included clinical, animal, agriculture, as well as wastewater surveillance. Our indirect surveillance activities include outreach and education, symptom monitoring of surveillance data for abnormalities. Priorities for national surveillance testing include the seasonal virus detection and characterization and detection of novel influenza infection. So the surveillance testing that we do at DCLS covers both. It allows us to detect seasonal influenza A and B. But with this testing, it would also help us to determine if we're seeing something novel based on the test results that we receive with our routine assays. But as Lisa mentioned, we're also doing sentinel surveillance. We are trying to use the roadmap that has been stipulated with the influenza right sizing initiative through the CDC, as well as supporting additional characterization and analyses of positive flu samples by using our National Influenza Reference Center. Just to talk a little bit more about these two initiatives, the National Influenza Reference Center, or NERC, is a state of public health laboratory that serves as an extension of the CDC's virology surveillance and diagnostic branch. They provide testing services to other public health laboratories that are complementary to CDC test methods. And so while public health laboratories in general are responsible for performing that first tier of testing using real-time PCR to identify flu A and flu B, as well as characterize the strains, the NERC centers will take it a step further. They will look at antiviral resistance using whole genome sequencing and other assays. They will also do the propagation as well as do the next generational sequencing. Participation in the NERC and having these centers implemented increased overall national capacity to do specimen processing, as well as enhance the efficacy of vaccine strain selections and detection of antiviral resistance. The National Influenza Right Sizing Initiative was implemented in 2010 by the CDC to systematically define the rationale, what critical capabilities should be in place, and optimize the right surveillance framework. It was determined prior to this that each state, each jurisdiction was doing things a little bit differently. And so this right sizing roadmap was a way to kind of define so that we can all be on the same page. The main objectives is to identify novel influenza, vaccine virus selection, antiviral resistance detection, and situational awareness overall of seasonal influenza burden. The goal of the first published roadmap in 2013 was to advocate use of new tools for molecular diagnostic testing, electronic laboratory reporting, and enhanced flu surveillance. The roadmap includes a standardized sampling strategy that is a representative of antigenic and genomic properties of those influenza virus strains that are circulating nationwide. This chart here gives a summary of the current influenza testing capabilities at DCLS. So as we mentioned, we are participating in the Sentinel Influenza Surveillance Program. We are also, in light of the HON that came out earlier this year, we stepped up and implemented capacity to prioritize accelerated flu A subtyping for those samples that are coming in from hospitalized patients where the flu A strain was not able to be characterized. And that could be either because it is a potential novel strain or because the testing that's done at the hospital does not have the capability to characterize the strain. And then, of course, we do testing for any suspected avian influenza, specifically H5N1. And then we also, prior to the Accelerated Flu A initiative, when we became aware several years ago that the molecular testing that tends to be implemented in hospitals aren't as sensitive in terms of detecting strains, we had already set up a process for hospitals to send samples to us that are flu A's that have not been able to be subtyped, and so that we can potentially detect any novel strains. Most of what we receive with this initiative are just either low virus titer samples that were not able to be detected due to limits of detection, or they have a test that's just not able to subtype. So these are our main initiatives when it comes to influenza testing. I'll also add that as a part of the Sentinel influenza surveillance, we are taking a subset of those samples and we are reflexing to a broader respiratory virus panel, which detects, I think, overall about 16 different respiratory pathogens, so that we can also take a look at other viruses that are a specific burden to our jurisdiction, and also giving us data on potential co-infections. Just to go into a little bit further about our testing capabilities, our main tests that we use are CDC developed methods that are FDA approved for diagnostic testing. These include the flu COVID-19 multiplex assay that will simultaneously in one reaction detect influenza A, influenza B, as well as SARS-CoV-2. And then we also have our various diagnostic panels for influenza that includes A and B detection, A subtyping, as well as flu B lineage determination. And then we have two specific assays, one that is specific for influenza A H5 and one that is specific for influenza A H7. The H5 and the H7 assays are not run routinely, only when we have been notified by EPI that there is a suspect case that could potentially involve either one of those strains. We employ these methods for our sentinel surveillance as well as diagnostic testing. As I mentioned, it affords detection of seasonal as well as COVID in one test, provides detection and subtyping capabilities. And of course, it will, with our diagnostic panels, it will very likely indicate a potential novel influenza strain if we are getting flu A detections, but we're not able to detect any of the seasonal flu strains. It could also give us some indications of variants based on the combination of flu strain analytes that are being detected in our assay. So our H5 response, it includes, as I mentioned, the diagnostic assay for the H5 subtype. We do concurrent testing for seasonal A and B as well as COVID, because as we know, the symptomology is all the same. So we do run those concurrently. We're accepting MP swabs, combined MP-OP, as well as conjunctival swab specimens in both viral transport media as well as universal transport media. Because we are dealing with potential novel strains or a strain that does not have a lot of immunity within the population, we do enhance our biosafety precautions when we do this testing. So initial sample processing is done in our BSL-3 containment laboratory with our biothreat emergency response group, which was a new group that was implemented at DCLS back in 2023. So this group handles all of our potential bioterrorism and newly emerging pathogens that require handling at a higher safety level. And then, of course, we do real-time PCR testing, and that expertise lies in our molecular detection and characterization group. We perform testing both during and after normal business hours based on health status, risk, and exposure assessments. We do have staff that remain on call to respond to these cases as needed. We also provide specimen collection kits that includes the swabs as well as the transport media, and if needed, also shipping materials. We do use the DCLS bonded carrier for routine transport as well as emergency dispatches as needed. And then, of course, we rely on VDA's testing approval and advance notification before we would move forward with testing in DCLS. And that is all I have. Thank you.
So for those who don't know me, I'm Dr. Carolyn Bissett with the Virginia Department of Agriculture and Consumer Services. I lead the Office of Veterinary Services there underneath the state veterinarian. And I'm going to talk a little bit about animal health. So we're going to switch it over to the stuff that I care about, which is animals. Not that I don't care about people, but I'm going to switch it over to animals for a minute. Before we get into surveillance, I just wanted to say that we are officially AI free in Virginia. So that's very exciting. We do not have the virus in, John may tell you differently when it comes to wildlife, but from my standpoint, the domestic poultry and cattle, we are AI free. We started this current outbreak, if you will, in 2023. We have had 11 detections here in Virginia in domestic poultry, whether that be a combination of, we've had 3 detections in commercial poultry operations and 8 in backyard operations since 2023. And as Dr. Darby mentioned earlier, this is unique in the length of time that this outbreak has taken. We're 3 years in, and we just had a detection in January in commercial poultry. So here in Virginia. So it doesn't seem to be slowing down, which is a little unique. The other unique factor is why we're all here today, and that we've seen 70 detections in people here in the United States since 2023, which is definitely unique. Now, we've had 1 death, and that person did have comorbidities or other health concerns. The rest have been mild disease, mostly conjunctivitis and some mild respiratory symptoms. So this remains largely an animal health disease incident. But of course, 70 detections over 3 years is still nothing to laugh about. So that's why we're here today. So I'm going to go into how we do surveillance for poultry and cattle, which not only protects them, but hopefully protects human health as well. So pre-movement surveillance. So all commercial poultry being moved within the state of Virginia, and this actually goes outside of the state of Virginia as well. This is part of National Poultry Improvement Plan, which most commercial entities need to be a part of to market their poultry, have to be tested for avian influenza within 14 days of movement. That includes birds going to slaughter, so they don't get a pass on that. Pullets or egg-laying birds going to layer operations, and finish operations. So basically any poultry moving, commercial poultry moving from one house to anywhere else outside that property has to be tested within 14 days of movement. So just to give you an idea, this is just Virginia numbers. That is about 30,000 tests that we did in 2024 on pre-movement. That's just Virginia movement testing. Again, I mentioned National Poultry Improvement Plan. This is a sort of a group, I'll say group's kind of an understatement, but a conglomeration of federal, state, and industry partners that have come together to form sort of policy-making, testing requirements, that sort of thing. And all commercial poultry producers need to be part of NPIP, again, to market their birds. And so there's certain testing requirements to be AI clean. There's also some other diseases you can be declared clean of as well. But of course, we're here talking about avian influenza. So again, commercial poultry companies are required to do routine testing in addition to that pre-movement testing. So for chicken breeders, and this really focuses in on our breeder birds, but about, what is that, 1,100 chicken breeders and about 600 turkey breeders. Just to give you an idea, which I should have said at the beginning, but poultry is our #1 economic industry here in Virginia. So it is extremely important for our economy. We are, I think, #7 in the United States for chickens and about #11 for turkeys currently. A little bit further down for egg laying. We don't have a whole lot of egg layers here in Virginia, but for chickens and turkeys, it is a huge industry here in Virginia. Backyard testing, so smaller backyard producers can enroll in NPIP as well. And this really benefits them if they're going to shows, fairs, selling product, that sort of thing. And then they'd be required to participate in routine testing for avian influenza. So you can see here testing a very pretty chicken at a fair there. Look at those feathers around the face. Kind of scary looking. He looks like one of those lizards kind of that does the, but very pretty chicken. And so you can see we do about 1,700 of those in 2024. Our staff goes to fairs and exhibitions on a regular basis to test birds there. So we sample birds at county fairs. It's a very busy time for us. Exhibitions. We're there at the state fair to test birds for avian influenza. Again, some of this is part of NPIP. Some of this is just part of what we feel is the right thing to do. And I think it protects public health as well to be testing these birds at shows where they're going to be interacting most with people. And so we did about 26 county fairs in 2024 and the state fair, and sampled about 1,200 birds. So live bird markets, this is a different sort of thing. And you can think of, if you've ever seen pictures, this is one extreme, okay? This is not how it happens in the United States, but if you've seen pictures of live bird markets in Asia, where there's just live animals everywhere, it's kind of that concept. This tends to be a cultural interest to certain cultures, but there are live bird markets here in the United States, and we have 3 in Virginia, where you can go, you can handle the birds, you can pick out which bird you want to take home dead, but you can handle it when it's alive. It is killed and processed there on site, and you take the carcass home for consumption. Very, very highly regulated here in the United States. So you can see those pictures from mainly Asia, other parts of the world, highly regulated here in the United States. But we do have 3 here, and part of the requirement to have a live bird market is to have routine AI testing. So again, that pre-movement testing, they have to be tested before they're moved to the live bird market and be negative. Then we make them close down every quarter, sell out all the birds, do a complete cleaning and disinfection, and then do environmental sampling before they're allowed to bring birds back. And that's done quarterly.
Question,
Yes.
where are those 3 located?
Northern Virginia. One's in Alexandria and the other 2 are sort of Loudoun and I think Prince William. So, and then we respond to sick bird calls. We get a lot of calls about from poultry, mainly backyard poultry owners who have some sick birds. A lot of people with backyard poultry don't have a regular veterinarian for their backyard poultry. So they'll call us when they have sick birds and we will take some information, determine if they qualify for, or not qualify, but determine if it's a high risk for avian influenza. There are lots of other diseases of backyard poultry, so not everything is going to need to be tested for avian influenza, but certainly if they are experiencing symptoms associated with that, then we will send staff out to do testing and test those within 24 hours. So you can see we did sampling about 135 birds in 2024. We also offer necropsy services that includes avian influenza testing. So if a backyard producer has lost several birds, they can bring one in for necropsy and we'll do testing for multiple diseases, including avian influenza. So let's talk about cattle. So as Dr. Darby and others have mentioned here this morning, this was a new twist to avian influenza in 2024, where it actually started in 2023, where some herds in Texas began experiencing clinical signs of mastitis and low milk production, and nobody could figure out what was going on. And some brilliant veterinarian said, well, why don't we test for AI just to see? And lo and behold, it was positive. So we started doing more testing, and it turns out that probably what happened was some wild waterfowl in a pasture with dairy cattle that were grazing. Somehow, we call it a spillover event, somehow that virus spilled over into cattle. And for whatever reason that we still don't quite understand, when it gets into these dairy cattle as a virus, it's concentrating in the mammary glands and in the milk. So it is being spread then from cow to cow through the milking equipment. The virus gets in the milking equipment, and even though that's sanitized and disinfected, it doesn't always kill virus. So it's kind of being spread that way. So in response to that, USDA issued a couple of federal orders. First, in April 2024, they issued an order that required all lactating dairy cows, if they're going to be moved between states, to be tested for avian influenza before. And then any positives would have to be quarantined. What we see in birds, and I'll talk about this at the end, is as we've already mentioned, HPAI, especially avian influenza, kills birds. Really, really high mortality, really, really high disease symptoms. What we're seeing in cattle is mild to moderate symptoms, mostly milk related, and they're not dying of the disease. So it's a little bit different management of the disease itself. And then in December of 2024, USDA issued what's called the National Milk Testing Strategy Federal Order. And basically they required all states to develop a plan to test their dairy cattle for avian influenza, and ultimate point of the plan is to be declared free of avian influenza in your dairy cows. So in January, we started testing, we being Virginia and VDAC started testing monthly farm level. Basically, we take milk from the bulk tank and we test that's already being collected for other regulatory purposes and test that for avian influenza. The good news is, again, here in Virginia, we are AI free. So we have, this slide is a little old now. Since January of 2025, we've tested over 600, close to 1,000, actually, milk specimens from Virginia dairy farms. All have been negative. We've tested over 50% of all of the grade A dairies in Virginia. And again, all negative for AI at this point. If a positive herd was detected, then of course we would quarantine that herd. They would not be allowed, the owners would not be allowed to move the cattle, move the milk. We would have to deal with, we'd probably still want the milk to be pasteurized because pasteurization kills the virus. But we would take some severe, rigorous biosecurity and management strategies on that farm. Again, I kind of covered this already. Again, we've already talked about this. So what would we do? I kind of talked about this a little bit. So how we deal with poultry and cattle, different. Talked about how we would deal with cattle, poultry. Unfortunately, the only sort of way we have to deal with a poultry detection, and especially in commercial poultry, but even so in backyard, is to depopulate those birds, both from a welfare and a public health standpoint. Again, these birds get really sick. I've got gross pictures if you're interested of birds looking really sick and dying from avian influenza. It's not pleasant to see. So from a welfare standpoint, that's the best thing to do. It also cuts down on people exposure and those birds moving and transmitting the virus. So for dairy farmers and poultry farmers, the best thing to do is practice good biosecurity. We mentioned that word already. That means having dedicated clothing, PPE, and footwear for each barn that you're going in, practicing good hygiene, recognizing sick animals, and then, of course, following testing protocols, surveillance protocols. And as of right now, as I mentioned again, we are AI free in Virginia. So let's just hope we kind of continue that way. All right, John.
Hey, I just have one quick question, and it's for selfish reasons, but how does VDAX monitor like the backyard flock swaps? So like there's a big swap tomorrow in Hanover. Does VDAX get involved in that at all?
If we don't. Okay, so if we know about them, and the reality is we don't know all of them, then we will send staff there to do testing just like we would for fairs and exhibitions. But the reality is we know a lot of them happen like in Tractor Supply parking lots. And we don't know about those. And that's just the reality of it. Is that kind of maybe not a satisfying answer, but an answer nonetheless?
Morning. We are not AI free in Virginia.
In wildlife. You have to specify.
In wildlife, yes. On the domestic side, we are. But I'll give you some updates from what we're seeing on the wildlife side here. So my name is John Tracy. I'm the wildlife veterinarian for the Department of Wildlife Resources. I just give you a very quick background. We have a really small wildlife health team within our agency. So it basically consists of two people, myself, and then we have Alexandra Lombard, who's our state wildlife health coordinator, and a handful of technicians that help us with, chronic wasting disease testing and some other things. But you'll see, I think, going through some of this, that we're a little bit resource limited in some of our response capacity for some of these big events. But this has been covered a few times already, but just to reiterate, so waterfowl especially, but waterfowl and some shorebirds and some gulls are really the reservoir for avian flu viruses. And these birds have low path viruses circulating in these populations all the time. So it's important to know that these low path viruses in these species are basically subclinical. They don't cause any type of individual or population level disease. And then high path viruses can also be asymptomatic in some of these species, but do have the capacity to cause disease. And so these high path viruses, that we're talking about, and now specifically the last few years, this H5N1 virus, we've seen a lot of birds susceptible, a lot of different species from a lot of different groups. And, why we see it in some species and not others, I think we're still learning, but we know the life history matters. So, for instance, birds that congregate heavily, like snow geese, can have major die-offs. We haven't seen huge die-offs in Virginia. We had, 30-some snow geese die on the eastern shore, but like Pennsylvania had 5,000 die in one quarry. So, like birds that concentrate at high densities can certainly spread virus. And then water also matters. And so just some species that we've seen disease in Virginia, Canada geese, snow geese, a number of shorebird species and other water bird species. So eared grebes and sanderlings last April took it on the chin, and then really our raptors are affected pretty severely, but more on an individual level and not so much on a population level from scavenging, or preying on infected waterfowl. So we've had a number of eagles die of high path. We've had peregrine falcons, great horned owls, barred owls, a number of hawk species. They're all pretty susceptible. And then black vultures have presented a bit of a unique case for us because starting in 2023, like basically all through the summer of 2022, there were huge mortality events in black vultures all over the state and really all over the eastern seaboard. And I mean, we're talking like involving, okay, I got 3 birds dead today. The next day I got 50. The next day I have 250, and then the roost is gone. And so, what's been interesting about black vultures, they sort of self-perpetuate this. So they roost in very concentrated places, sort of at levels on top, and they'll even, they'll like defecate on each other as it goes down. And then if they get birds that die, they'll actually self-scavenge carcasses. And so they're sort of amplifying the virus within these populations. But those have been like what happened sort of in 2022, and this was really prior to me even being in Virginia. Those have been the sort of the most widespread wildlife mortality events that we've seen. But we have at times had various pockets of disease pop up in a number of species. And I'll show you kind of a little bit what's happened the last few months for us. All right, this has been shown also a couple of times, but I just want to point out a few things. And I don't think the laser pointer is working, but the initial introduction of this H5N1 virus was into Eastern Canada in late 2021. And so, it presumably with a bird that came over from, a flyway in Europe, or from a bird that commingled with birds over there and then sort of crossed the Atlantic and came down. And we also know that really since then, there's been at least 3 introductions into Alaska. And so if you look at Alaska on this map, you'll see why we're particularly concerned with virus spillover across continents in Alaska. So we have basically, all of these Asian flyways and all these North American flyways, and then they all sort of intermingle in Alaska. And so there's a real focus nationally on surveillance in waterfowl in the Pacific flyway. But interestingly, with this H5N1 virus, the initial introduction was on the East Coast. And just to bring this home a little bit, I'll give you a couple of examples from Virginia. So these are birds that are outfitted with little telemetry backpacks that can ping cell phone towers as they go past. And so you can see on the left, these are Canada geese, Atlantic population of Canada geese that were outfitted up in northern Quebec. And then, they kind of disappear, right? There's no cell phone network up there. So they sort of disappear. And then as they migrate south, they start showing up on the map. But you can see like the geese, the actual, Atlantic population of geese that we have in Virginia, how many states, they cross over and commingle, and some of them, even switch flyways and end up in the Midwest. And then on the right, these were black ducks that were actually banded by our staff as part of a American Black Duck project. So these were all banded in Southeast or Eastern Virginia, and you can kind of see the variety of places that they end up. And so talk about a One Health virus, right? We have this virus that has a reservoir that can move all over the world. It also affects domestic species and humans. And here we are, we're all sitting in the same room talking about this. All right, so wild bird surveillance. There's a few different things that take place routinely. And so I work for a state agency, but I really need to talk about some of the stuff that our federal partners are doing. And so this is a federal document, put together, basically a multi-agency document that laid out some strategies that could be employed for the surveillance of influenza viruses in wildlife. And so they laid out these 5 things, right? Investigation of morbidity, mortality events. I'm going to tell you a little bit about that because that's really what we do at the state level. Surveillance in live wild birds, primarily tacked on to research projects or population monitoring, banding events, those types of things. And then a big one that USDA does as part of their program, I'll talk about a little bit, is surveillance in hunter-harvested birds. And then they also lay out in here sort of the use of sentinel species, mentions of backyard flocks or even, strategically placed susceptible duck species and environmental sampling. But I'm not really aware personally of those methods being used at least in Virginia. Really, these first 3 are the bulk of the surveillance that gets done in our state. And so the testing strategy for these birds, these all have to be run through, a NALM lab, so a National Animal Health Laboratory Network branch. And the initial testing will be done with this matrix gene RT-PCR that's going to pick up all those influenza AIs, and then they'll move to specific H5, H7 genes, and then eventually, like anything that pops there is going to end up at NVSL for confirmation. And sometimes, this is not Carrie's fault, but sometimes the wild birds at NVSL don't get prioritized as high as the domestic counterparts. So sometimes we're awaiting confirmation for a while. Okay, and then so this active surveillance program, so this is really performed not by us at DWR, but by USDA, APHIS, Animal Plant Health Inspection Service, Wildlife Services. And so they have a national wildlife disease program that's based out of Fort Collins, Colorado. And they put together this surveillance goals and targets for the surveillance of healthy hunter-harvested ducks in various watersheds all over the country. And, there's a lot of epidemiological modeling that goes into this on where we think spillover might occur, where previous viruses have been isolated. And that modeling is honestly way over my head. But what comes out of this every year is that our local wildlife services staff are handed targets. So this was like the 2024 to 2025 targets, and there's a spatiotemporal relationship in this. So we want this many birds from this time, from this watershed. And so what they're doing in this case is they're just going out to boat ramps or, making connections with various hunt clubs. And these are all healthy, apparently healthy, dead now, but healthy hunter-harvested wild ducks. And so they'll get retropharyngeal anticoagual swab and then run those through their NOM network. And this is happening every year. The numbers change a little bit every year based on the modeling, but there's a plan and a target in place for every state. And so you can kind of see Virginia's not as high as in some of the states in that Pacific Flyway that we talked about, but certainly us being a seaborne state with the Atlantic Flyway, where Wildlife Services is asked to secure a number of samples that way. And so this is really active surveillance that's taking place every year, all over the country, every state, every year. And that's a little bit different than what we at DWR do. And so we can certainly contribute, to surveillance when we're handling birds, as I said, for research purposes or banding purposes or something like that. We can tack on surveillance for that. But really the bulk of the surveillance that we're doing is just opportunistic in response to reports from the public of clinical, or sick so sick or dead wild birds that may be suspicious of AI. And this isn't uniform. I mean, like we could probably go years and respond to a handful of cases, but then when we actually get high path, we've responded to like 300 in the last few months. So it's, it can be really taxing on our staff to be able to actually respond, and we have to kind of pick and choose where we feel like we're going to get the best epidemiological evidence, where we really need diagnoses versus what our capacity is. And so just, I have a couple of pictures over here. Like these are just examples of calls that we get from the public. So the laser pointer is not working, but these brown pelicans, this is down in Virginia Beach. Like, there's a handful of brown pelicans. I don't know if you could see, there's about 6 dead ones laying behind them, sort of scattered around. And so, we'll get a call like, hey, there's pelicans laying all over the place. And we'll make a decision really like, ooh, if we haven't had active, AI, if we don't have a readily, easily explainable cause for that, then definitely we'll go out, we'll collect carcasses, and we'll get a diagnosis. And so for us as an agency, we actually work a lot with a lab down at the University of Georgia called the Southeastern Wildlife Cooperative Disease Network. So SCWDIS is what it's always called. And it's really cool. So all these state wildlife agencies, there's 17 state wildlife agencies in the Southeast, a little bit in the Midwest. It's a true co-op. We pay an annual fee every year to fund this lab at the University of Georgia, and they do the bulk of our diagnostic work. And so, same thing, like we'll send birds down there, they'll get full necropsies, including histopath and variety tests. And then, anything that needs confirmatory testing, then they can send up to NVSL from there. And then, so that's a little bit easier, like, oh, there's a whole bunch of Pelicans Lane, we should figure out what that is probably. But then you'll get lots and lots of reports of like, hey, I found a dead bird. Like I have a dead bird laying at my bird feeder. I probably honestly can't respond to that. And honestly, it's probably not worth our effort. We really want to think in terms of population. Like this bird, we got a report, it's a single bird. It's a merganser, but honestly, I responded to this one because it's a waterfowl species and it was in a county in the western part of the state where we have had no prior cases. And so it was like, well, let's rule that one out. Whereas if I have a single bird laying, on the eastern shore in the midst of what I know is a high path outbreak, I don't need further diagnostics to know that that's high path. And even if it's not high path, it doesn't really matter. So we have to kind of like, because of our limited resources, we really have to like evaluate all of these on a case by case basis and try to determine, what needs diagnostics and what doesn't. And that's not always easy for the public to grasp. If somebody finds a bird in their backyard, they want to know if that's high path. But it's what we can do. And honestly, we're trying to provide the best, use our resources to provide the best epidemiology that we can. And so, since 2022, since this H5N1 virus emerged in 2022, we've had kind of waves of disease. And we had pretty widespread, like in the spring and summer of 2022, we had really widespread disease, especially in vultures. And then we're kind of quiet, and then we get a little spike in the fall with some, migratory birds coming through, maybe a little spike in the spring. But then last spring, so December, January, I say spring, winter, spring, December, January, February, March was like totally quiet. So 2024, early 2024, totally quiet. And then we got a blip, a really isolated mortality event involving grebes and sanderlings down in Southeast Virginia. It lasted like 3 weeks and it was done. And then it was completely quiet. No AI cases through the summer, no AI cases through the fall. And then some of our states to the north in December started reporting some mortality in snow geese. And then it was like second week of January, it was like you flip a light switch and we had birds all over, started on the eastern shore, sort of in conjunction with the commercial detection that they had, and then moved down the eastern shore, and like basically all of coastal Virginia was affected, and then kind of moved up into northern Virginia. And you can sort of see, so just putting the cases up here, and I don't have case numbers for how much we did, but just geography. This is where we've had virus on the landscape since early January. And I think this kind of geography can really help inform some of the public health decisions that you guys might need to make in terms of risk and determining exposure and things like that. So I think that's all I have, unless there's any questions.
I just have one question. For like the Southwest and like the Shenandoah Valley area, do you think that it's lack of reporting by the community? So I know for COVID that like Bath County and some of those more rural communities, they went COVID free for so long because people weren't getting tested. It was just underreported. Could that be the same situation here?
Well, potentially. So certainly we get more, just the density of people and the cultural differences, like we get more reporting from Eastern and Northern Virginia. But, with the nature of this disease, like it's not like we're missing one spotty case here and there. So like if we get reports from counties, like we're always getting multiple reports, sometimes of the same event. And so So I think that like if there was significant morbidity or mortality anywhere in the state, we would hear about it. We might not hear about it to the same intensity that we hear about it in other places, but I don't think we're missing it, especially not if there's, if it's there in any appreciable level. Now, like certainly I know Carrie says they just assume that, the virus is everywhere, that it's on the landscape. And it wouldn't surprise me, if we found this virus in any particular species anywhere in the state. But we have tested birds, so this just shows like suspected and positive cases. It doesn't show like where we've tested and had negative birds. And so we've routinely tested a number of reports in the Shenandoah Valley, especially with the poultry industry up there, and those have all been negative this latest round anyway. Yes.
Of course, I'm partial to anything lab related. Can you talk a little bit about the confirmatory testing that NVSL performs?
I think I can. So all of the suspected cases need, because it's a foreign animal disease, must get confirmed at NVSL. And so NVSL will, they'll run through the standard PCRs to get confirmatory testing. But then what's nice is that they'll do all the subtyping and genotyping really. And so that's where, they like, if we know we have D1.1 on the landscape or something different, like that's where we're getting all that information from. The thing is that with what's been going on, so all the domestic testing and the dairy testing, and then like sort of widespread mortality events in wildlife, like the lab is just totally swamped. And so I actually don't have confirmatory testing from any avian species yet that has been tested in this calendar year. And I might not get them till like this fall. So they're just not highly prioritized. They are prioritizing mammals though, because I didn't mention this, but we've had 2 red foxes test positive, both in Loudoun County, which were the first mammalian cases in Virginia. So it wasn't surprising. I mean, red foxes have been diagnosed in a number of states, but we just haven't seen it in Virginia. And NVSL got me those, at least they put them in a clade. I didn't get the genotype yet, but they put them in a clade and got me those like within a week. So there is some priority there. And we don't submit directly to NVSL, so that's all coming through the National Animal Health Lab network. So
I've got a question regarding birds testing positive and or negative. Are there cases of birds that are testing negative? And if so, are those being set aside as because they may have some type of immunity built up?
Yeah, great question. So we do have a number of birds that have tested negative, and what we've been doing, especially with raptors, is, if I receive the carcass, we may just swab it and freeze back the carcass and just initially get the flu testing done, and then if it tests negative, we have the carcass, we can go back and look for further diagnostics. The population immunity question is interesting because there hasn't been a lot of post-mortem, immunity work done, but we are looped into a project with a flu researcher, Becky Polson down at Squidus, looking at population level immunity of eagles. And so we've been handling a lot of live eagles and providing, paired serum and swab samples for those. And it's been pretty interesting. Like we handled 6 eagles up on the northern neck, basically at the same time or within a few weeks of when we knew there was high path on the landscape. And 4 of those 6 birds have antibody. And so they're working to characterize the antibody profile because certainly population immunity has to play a role in some of this.
Exactly. That's the way that I was thinking as well. That if there is antibodies created by the birds, maybe have those being bred to ensure that the population survives.
Yeah, so it's the work that we're doing with eagles is sort of opportunistic. It's a small project that we set up with Becky, but also I know that on the USDA side, there is a lot of serological work done in waterfowl as well. So, in addition to the PCR work that's looking at live virus, they're also looking at serological profiles of live ducks up and down sort of throughout the country to pair that with serum and get a gauge of, that goes into the modeling on what kind of a wave might we expect with this fall's migration. If we know have an estimate of what the population immunity might be, then maybe we don't expect there to be such a widespread viral replication event or more morbidity mortality event.
Thank you. And I was also thinking about the turkey and chicken population that we used to consume as far as breeding goes.
Yeah, I might have to let Carrie speak to that one.
Yeah, USDA Veterinary Services, Agricultural Research Service is working on doing some research. I'm not sure where they are in that on breeding influenza resistant poultry, domestic poultry. So I'm not sure where that is. It's just in this with the new administration been one of their 5 viewpoints to do research on that. So I think the research is just beginning. So we're many years away from that, and how successful that will be, I don't know, because as we mentioned earlier, influenza mutates and shifts so frequently that I'm a little skeptical that will be successful long term. But maybe it is a tool that would get us out of the current outbreak situation.
Thank you for speaking on that.
I guess just lastly, I should say that, and maybe I shouldn't say it because I'll jinx it here, but we are on a downward trend. Like we've been swamped with AI calls and reports of sick and dead birds since the middle of January, and it has really started to taper off now in the last few weeks. So hopefully we'll stay quiet for a while.
I just have one more question then, because the last few weeks have been pretty warm. Is it based, if it stays relatively hot, we have a pretty hot summer, will that affect the fall potentially?
Potentially. So influenza, a cold loving virus, right? So it definitely through the summer, we wouldn't expect as many cases as we would maybe other times of the year. But I don't think it's all heat. I think it's probably a combination of environmental conditions like temperature and also which birds are on the landscape. And so these, in the fall and in the spring, we have like just incredibly dense populations of waterfowl that are migrating through. And so just because even if it's really hot and we don't have a lot of viral persistence through the summer, that doesn't mean that the fall isn't going to bring a wave of infected birds from the flyway. Thanks for having me today. Appreciate it.
Use telemetry backpacks are cool. We do not have that. Gonna shift back to human surveillance a little bit. And Lisa covered a lot of these steps, and I realize we're getting short on time, so I'm going to try to move through here pretty quickly. These are the basic steps in a public health investigation, and I'm just zooming in on that process and giving you guys a couple of examples from some rule-out investigations that we've had in Virginia. And most of these steps that I'm going to go through would be undertaken by a local health department, so a district epidemiologist in partnership with a communicable disease nurse and their district team, and also with our regional epidemiologists. And they would start these actions if there was a lab result that was a flu A positive, but was not either H1 or H3, so a seasonal strain. Because of the potential, as you guys have heard, for sustained person-to-person transmission and a possible pandemic, we take investigation of a single unsubtypable event in humans take it very seriously as a public health priority for investigation. And these are the main on this slide focuses of our public health investigation. So that falls into three main categories. One is to identify what is the source of infection? How was the person exposed? Two is person-to-person transmission of virus occurring. And then three, probably most importantly, we want to slow or stop the spread of virus in people to reduce morbidity and mortality to disease. So we're going to take a closer look at each of these three areas and walk through what activities might be conducted around both the initial person who's ill, so a suspect case, and then the people around that person, so the close contacts. So first, a public health investigator would have this suspected case of disease reported to them, and they would ask them questions about travel, about exposures, about consumption of animal products, and about potential contact with any other person who had an illness that could be due to influenza. So they would start by asking them questions. Have you traveled within the United States? Have you traveled out of the country? What did you do while you were there? Who did you go with? Is anyone else ill? What did you have contact with while you were traveling? The investigators would also ask about any potential exposures to animal sources that carry influenza. You heard about wild birds, poultry, cattle, swine, and other animals. But we would also ask if they attended an agricultural fair. Did they have backyard chickens? Did they go to a petting zoo? And did they handle any animals there? As well, we would ask about exposure to animal products. Like, did you consume any unpasteurized milk or yogurt or cheese that could have contributed to your infection? And I mentioned also early in an investigation with a single untypable result, we would ask them if they had had contact with any other ill persons, thinking that maybe that other ill person didn't get tested and could have possibly been a source of their infection. Some of the considerations that field staff often encounter are assessing whether they can interview the person over the phone or whether they might get better information from an in-person visit. So during a recent investigation where a novel subtype was being ruled out, we did have one of our investigators travel to the person's home. They had been hospitalized, they were discharged home. We just were notified about the case and they had been fit tested and had proper PPE. So in that situation, they felt comfortable going into the household to interview the person. And it also enabled them to collect a follow-up specimen on the person. In public health, we usually use a case report form to guide our investigative process, just to make sure that we're asking consistent questions throughout. And we have a VDH case report form, and there's also a CDC case report form that we used. Sometimes during the investigation, we might find that the person who is ill can't be interviewed. That could be because they are very sick. Maybe they're in an intensive care unit or they're on a ventilator. Maybe the suspect case is a child or an infant or a toddler who can't be interviewed. So in those situations, we occasionally interview a proxy, such as a spouse or a parent, to collect that important information. Okay, so back to our case investigation. The things that we want to know about a person are: what symptoms do they have? What's the clinical course of illness? What underlying risk factors do they have that might make them more prone to more severe illness manifestation? And then also inquire about their vaccination status with the current seasonal flu strain. So use an example from Virginia that we had where we were reported with an initially unsubtypable result. This suspect case was in an adult male. He had no domestic travel, no international travel. The only animal exposures that he had were backyard chickens on his property, but he said that he did not have any interaction with the backyard chickens that were well and not sick and no die-offs. For underlying illnesses, he did have metastatic cancer and had been hospitalized for multiple days and then was in the recovery process by the time we found out about it. So moving back to our investigation, after getting some initial information from the case, we want to determine, is there person-to-person spread occurring? So here our goals are, one, to determine if another person could be the source of that individual's infection, and identify close contacts that are around the suspect case since they became infectious to determine if there is possible spread from them to other individuals. I think Lisa defined the close contacts. We look at individuals who were within 6 feet of the person for a fairly prolonged period of time, or anyone who has had direct contact with infectious secretions while the patient was likely to be infectious. That could be someone who got sneezed upon or someone who shares cups and utensils at home, or it could be a healthcare worker at a facility who didn't use the proper infection control precautions. So in the beginning, we would cast a really wide net around the person. You have a sick individual and we want to look at, does the person work? Where are they in the community? Who's in their household? To get that big list going. And then as we get deeper into the investigation, we really ask about the nature of the contact with the person to determine whether they might be a close contact. For our suspect case that we worked with, he had when interviewing him, he had an exposure to an ill family member who traveled internationally, and also a visitor to the household who had traveled internationally. So we were looking at them as potential sources of exposure for our suspect case. And then after he became infectious, we look at individuals who he could have passed the illness onto. So close contacts in the household that visited frequently. We identified about 5 to 7 individuals who either lived in his household or were visiting frequently enough to be considered as close contacts. And then there were maybe 12 or more healthcare workers who treated the patient before proper infection control procedures were undertaken. So important public health goal outlined here is to slow or stop the spread of infection. We want to ensure that we're taking active steps to reduce the spread of disease. One of those methods we can do is to initiate antiviral treatment with the case, so that can reduce viral shedding and reduce the likelihood of spread from the case to other individuals. We also want to make sure that proper infection control procedures are undertaken. So both at a healthcare facility, which Carolyn will talk about, but also at home. Our case I mentioned has been discharged home by the time we found out about him. And so we really wanted to educate those in his household, particularly his caretakers, and make sure that they had masks and gloves that they needed when they were interacting closely. Okay, and then for the close contacts that we identified, we enrolled them in what we call symptom monitoring. It's not as cool as backpack telemetry, but we work with the close contacts and we tell them, please measure your temperature twice a day, be alert for onset of fever, cough, sore throat in the next 10 days, and monitor yourself. And we want to make sure that we identify them very early so that they can be tested and isolated away from others and, if needed, treated. So that was the sort of end of our slides here, but I just wanted to mention that in our case study, none of the close contacts that were identified developed any symptoms of illness. We did provide antiviral medication, preventive treatment to some of the high-risk contacts because they were in the household or had had aerosol exposures. And then ultimately, in our lucky situation, the initial unsubtypable result was determined to be a seasonal strain after it got down to the CDC for further testing.
And so here I'm going to pass to Carolyn Kiefer, and Carolyn is online. So Carolyn, can you do a mic test to see if your audio works?
Hi, all. Can you hear me?
We were hearing you possibly very faintly, but is the audio in the room, can that be turned up?
Can you hear me any better?
It's a little better. Can you try again?
Yes. Hi, good morning.
Great. All right. And we can advance your slides here if you just say next slide.
All right, perfect. Wish I could be with you all this morning, but saying hello from beautiful Southwest Virginia. So I'm Carolyn Kiefer, the Healthcare Associated Infection Antimicrobial Resistance Nurse Epidemiologist with the HAI-AR program within the Office of Epidemiology. So with what Don covered with the investigation, there's what we learned about the impacts and key measures of infection control, patient management, and healthcare worker safety, and then post-exposure management. So with healthcare planning, it's important to understand the unique infection control challenges with managing patients with possible novel influenza A viruses, which then creates a domino effect. This is based on what we learned firsthand. So if you remember anything I covered, this is the most important slide because we need to understand the challenges to be able to put the preparedness into context. So the challenges start with delayed identification and implementation of the right infection control precautions if there aren't necessary screening and testing in place, or if there are breakdowns in established processes. It's important to note that the recommended precautions are airborne and contact, which are far more rigorous than precautions for seasonal influenza. Sorry, I just lost my spot. So because of the delays during that time frame, there are going to be ongoing healthcare personnel exposures occurring. So then when a case is finally suspected, there's a challenge of contact tracing and identifying who may have been exposed, which can be many, possibly hundreds from what we observed in our investigation. And then it can really depend on that time frame and the healthcare that's provided as well. So being able to quickly identify those at risk for infection, especially those that are higher risk, such as certain healthcare personnel with health conditions or involved in procedures like aerosol generating procedures, AGPs. And then additionally, considering how you're going to identify and manage any exposed patients and visitors. So with implementing post-exposure recommendations, there can be further challenge with after hours availability of occupational health to be able to support personnel follow-up, and then with managing exposed personnel, meeting the CDC's recommendations to furlough those exposed during that 10-day monitoring period, or choosing to allow them to work after testing and providing chemoprophylaxis, which I'll cover more in the next slides. So with all these challenges laid out, the first and most important line of defense to prevent them is early identification. Preparedness plans must include screening protocols implemented at all entry points, and that includes questions on symptoms and exposure history. Screening should ideally start before a patient's arrived to prevent any exposures, and then as soon as possible at arrival to the facility, and then during their healthcare visitor stay. Symptomatic patients identified to meet all the screening criteria should immediately be instructed to follow respiratory hygiene practices and be masked. And then healthcare personnel should be implementing standard contact and airborne precautions. That means wearing gloves, gown, eye protection, and at minimum a fit tested NIOSH approved disposable N95 respirator. Then also to note that personnel entering that isolation room should be limited and tracked. Healthcare personnel should be dedicated to only caring for that patient to minimize any transmission. And then patients should be placed in an airborne infection isolation room if available, or the private room with the door closed. And also using any other engineering controls appropriate to the situation to help continue to reduce or eliminate any potential exposures. Next. So in the event of a suspected case, antiviral treatment should be provided as soon as possible, not waiting for lab confirmation. The healthcare facility should communicate to their local health department, who then can assist with lab coordination for further testing, and of course, expediting to DCLS for subtyping of all influenza A positive specimens, which Dr. Griffin Thomas covered all of the lab aspects in her previous slides. Next. Environmental infection control is always essential for infection control, and healthcare facilities should ensure there's thorough and consistent cleaning and disinfection using EPA registered disinfectants with label claims that are effective against avian influenza viruses. Observing actual practices is recommended to make sure it's done correctly and effective. And that can be done using some of the CD's observation forms. There should also be processes for monitoring visitors and managing those visiting patients in isolation. They should be provided training before entering the room, such as with hand hygiene and personal protective equipment, PPE. They should also be instructed to limit their movement within the facility. And then if they do develop any signs and symptoms, that they see their healthcare provider, and they should not visit if they're ill. And then the facility should have processes in place to monitor severe respiratory infections within their facility and encouraging healthcare personnel to self-report acute respiratory illnesses. Next. Communication is always important, and it should include strategies to keep healthcare workers informed and prepared. This includes communicating about any increased respiratory illness activity, or if there's an outbreak detected within the facility, and then providing education relevant to personnel's jobs or tasks so they can be able to know what they need to do to prevent any disease transmission, such as with AGPs. And then again, ensuring accountability with key infection control practices through monitoring adherence, like for transmission-based precautions when caring for symptomatic patients. And CDC does have another audit tool for that. Next. So now let's get into what it looks like if appropriate precautions were not used and what we do to evaluate healthcare personnel exposures in accordance with the CDC protocol. The rationale behind healthcare worker exposure protocol is why it's stringent because of the disease severity and the risk for high mortality. So according to the CDC definition, unprotected exposure is defined as being within 2 meters of a symptomatic patient with novel influenza A virus infection, and they were not wearing the recommended respiratory and eye protection. So the recommended PPE that CDC points out is a minimum of N95 respirator and goggles or a face shield. So back to the challenges we previously covered, many healthcare personnel evaluating a patient with respiratory symptoms may be wearing some type of respiratory protection, most commonly a face mask. So they're usually not wearing an N95 respirator as recommended in the guidance, unless this facility does have those stringent screening protocols in place to address novel influenza A. So in addition to a challenge, eye protection is worn less frequently, and use of respiratory and eye protection is difficult to verify retrospectively, so it's not usually documented in the medical record. This increases the number of healthcare personnel who could be considered to have an unprotected exposure. So for these exposed personnel, they should be excluded from work until 10 days after their last exposure and monitored for signs and symptoms during that period. But fortunately, CDC does provide an option if furloughing staff for the 10 days is not possible to ensure there is adequate staffing of the facility. So these asymptomatic healthcare workers could be considered to work if all of the following occur, which is having a negative influenza test, are started on post-exposure antibiochemical prophylaxis within 2 days of the exposure, and then they wear a face mask for source control during that 10-day post-exposure period. Next. Oh, you're there. All right. So, but due to what we've learned for the known practical and logistical challenge associated with that strict adherence to the current CDC guidance, VDH does offer an alternative option that balances safe and effective patient care with realistic staffing and operational needs. So in lieu of furloughing asymptomatic exposed healthcare personnel, this alternative option does include adherence with all the following. So having that active monitoring by the public health or the healthcare facilities during that 10-day period, using universal source control, monitoring compliance with IPC practices, so hand hygiene, respiratory hygiene, etc. Focusing on those units where the exposures occurred and where the healthcare personnel worked. And then in consultation with VDH, could consider testing of asymptomatic healthcare worker if feasible It could be those focusing on a higher risk personnel, like those with AGPs. And then providing chemoprophylaxis as soon as possible to healthcare personnel with those higher risk exposures. Again, could be those AGPs, is what we've learned. And then those with those underlying medical conditions that make them more at risk for any flu complications. So anyone aged 65 and over could have asthma, diabetes, etc. And then lastly, for any healthcare personnel who do develop symptoms, they would need to be started on any antiviral treatment. Next. So what about those healthcare personnel that provided care to an ill patient, but did wear all the appropriate PPE and followed all the recommended processes for putting it on and off? So we know there can be unrecognized failures in PPE practices, so we would still want to monitor those asymptomatic exposed personnel, and that should be during that 10-day period as well. Next. So if healthcare personnel do become asymptomatic, they need to immediately report that to a supervisor, isolate at home, wear source control, and practice strict respiratory hygiene around others in the home. There also need to be some coordination with medical providers to be able to make sure they get prompt evaluation and treatment. And then as a reminder, your early antiviral treatment is key, especially those that have those underlying health risks. So next slide. So some BDH and CDC references of the content I addressed. So that sums up the healthcare facility key considerations, and I will hand it back over.
There was a question in the chat from Mark Kilgore. Do federal authorities and customs report to state like VDH foreign travel on farms?
Not a question I would have an answer to. Is that something Don or somebody else from VDH that might have an answer?
Can you repeat the question?
Yes, it says, do federal authorities and customs report to state like VDH foreign travel on farms? And Mark, if you're on to elaborate that question, you can speak it.
Not that I'm aware of. I'm not aware that they've reported to us any travel abroad to farms.
Yeah, no, I'm not aware of that either. What I would say is if there was an affected farm, we would be doing on-site epidemiology work to identify contacts. So that would include anyone there, regardless of their status. Oh, abroad and then returning here. No, that's a good question. I have not heard anything about that.
Yeah, that's a real common question. I travel a lot to Latin America. You know, they're very sensitive about bringing in fruits, of course. They don't want to transmit plant viruses, let alone animal viruses. But yeah, it was sort of like if they're sharing that information to you. I know I've been in conferences before where we've had international travelers, and there was an Ebola concern. And I believe this, I can't remember if it was, because it's been a few years, if rather federal or state health officials came to the conference and then interviewed the international attendees from the affected countries, because they were trying to stop the Ebola vectors or whatever if they were present. So I was just curious on those kind of issues on federal state cooperation in this sort of battle against human and animal and plant disease. So thanks.
It's a good question. We do work closely with CDC Division of Global Migration Health, and they do during certain periods when there are Ebola outbreaks in Africa report those who have been to affected countries and had certain exposures. They do share that information with public health, and we reach out to them and sort of enroll them in health monitoring and give them the names of contact information for local public health. But I'm not aware of any information we've received about travelers to farms abroad, but thank you.
I appreciate it, but I won't apologize because it was all good and necessary information. As we noted in our intent this morning, it was to frame the threat for you, to set the base for the educational base for where we go from here. So in the chat and on the screen, there is a survey that we came up with in in OEP, and it's just basically an opportunity for you to put things. There's a couple questions related to continuity, lines of succession, delegation of authority, some things we learned in COVID. Plus, there's also a free form place if there's any other training or elements you want to see added to or in advance of BIO100, which my intent is to kick the planning off for that this summer. And we'll be reaching out and putting a team together for that, because as you can see, we just framed the threat. You can see how many moving parts and how many folks need to be involved that weren't. This is just as it's happening. So, and it will be operational. The BIO100 will be operations based. We'll be opening dispensing sites. We will be moving simulated material around the state because that's a requirement. So that will be sometime next year. We'll set the date once I get the planning team put together. But again, thank you to all of our presenters. I appreciate you all jumping in and being here to help us frame this for everybody. And that's all I've got. Jennifer, any parting thoughts?
No, just thank you, VDH. Thank you, VDAX. Thank you, Wildlife Resources, and everybody for very informative and great information today. It was very intriguing and understanding all of that. Just if you will, remember to do the VEST training attendance. I think Franklin had just put it back in the chat. We appreciate you all hanging with us a little bit later today. But again, as Aaron stated, it was really well worth it. So appreciate it.
Two more quick things I forgot. I'm sorry, I'm getting old. I will publish an executive summary as documentation of this event, and we will disseminate that through the VEST mechanism as we normally would. And then we're also going to take this recording and I'm going to put it in our train system when I get back to VDH, and we will send that link out. So if you wanted to run this for an in-service or you wanted to use